Bismillahirrahmanirrahim. This video is going to explain the various processes which replenish or regenerate ATP molecules in muscles for a continuous supply of energy. Before watching this video, you must watch my video on the cross bridge cycle. The link of that video is available in the description. Let's start if you have already watched that video. Adenosine triphosphate is the energy for muscle contraction. In addition to its direct role in the cross-bridge cycle, ATP also provides the energy for the calcium pump in the sarcoplasmic reticulum, which is an example of the active transport. Muscle contraction requires large amounts of ATP. The amount of ATP stored in muscles is very low, only sufficient to power a few seconds worth of contractions. As free ATP is broken down in the muscles, it must therefore be regenerated and replaced quickly to allow for sustained contraction. There are three mechanisms by which ATP can be regenerated in muscle cells: creatine phosphate metabolism, anaerobic glycolysis, and aerobic respiration. Let's discuss these mechanisms in detail. Creatine phosphate is a molecule that can store energy in its phosphate bonds. In a resting muscle, excess ATP transfers its energy to creatine, producing adenosine diphosphate and creatine phosphate. This acts as an energy reserve that can be used to quickly create more ATP molecules. When the muscle starts to contract and needs energy, creatine phosphate transfers its phosphate back to ADP to form ATP and creatine. This reaction is catalyzed by the enzyme creatine kinase and occurs very quickly. Thus, creatine phosphate derived ATP powers the first few seconds of muscle contraction. However, creatine phosphate can only provide approximately 15 seconds worth of energy, at which point another energy source has to be used. As the ATP produced by creatine phosphate is depleted, muscles turn to glycolysis as an ATP source. Glycolysis is an anaerobic process that breaks down glucose to produce ATP. However, glycolysis cannot generate ATP as quickly as creatine phosphate. The glucose used in glycolysis can be provided by blood or by metabolizing glycogen stored in the muscle. The breakdown of one glucose molecule produces 2 ATP and 2 molecules of pyruvic acid, which can be used in aerobic respiration or when oxygen levels are low, converted to lactic acid. If oxygen is available, pyruvic acid is used in aerobic respiration. However, if oxygen is not available, pyruvic acid is converted to lactic acid. This conversion allows the recycling of the enzyme nicotinamide adenine dinucleotide, which is needed for glycolysis to continue. This occurs during strenuous exercise when high amounts of energy are needed, but oxygen cannot be sufficiently delivered to the muscles. Glycolysis itself can only be sustained for approximately 1 minute of muscle activity, but it is useful in facilitating short bursts of high intensity output. This is because glycolysis does not utilize glucose very efficiently, producing a net gain of 2 ATPs per molecule of glucose and the end product of lactic acid. The lactic acid may contribute to muscle fatigue when it accumulates in the muscles. Aerobic respiration is the breakdown of glucose or other nutrients in the presence of oxygen to produce carbon dioxide, water, and ATP. Approximately 95% of ATP required for resting or moderately active muscles is provided by aerobic respiration, which takes place in mitochondria. The inputs for aerobic respiration include glucose circulating in the bloodstream, pyruvic acid, and fatty acids. Aerobic respiration is much more efficient than anaerobic glycolysis. Aerobic respiration produces approximately 36 ATP molecules per molecule of glucose versus 4 from glycolysis. However, aerobic respiration cannot be sustained without a steady supply of oxygen to the skeletal muscles, and it is much slower. To compensate, muscles store small amounts of excess oxygen in myoglobin, allowing for more efficient muscle contractions and less fatigue. Aerobic training also increases the efficiency of the circulatory system so that oxygen can be supplied to the muscles for longer periods of time. Muscle fatigue occurs when a muscle can no longer contract in response to signals from the nervous system. The exact cause of muscle fatigue is not fully known, although certain factors have been correlated with the decreased muscle contraction that occurs during fatigue. ATP is needed for normal muscle contraction, and as ATP reserves are reduced, muscle function may decline. This may be more of a factor in brief, intense muscle output rather than sustained lower intensity efforts. Lactic acid buildup may lower intracellular pH, affecting enzyme and protein activity. Imbalances in sodium and potassium ions level as a result of membrane depolarization may disrupt calcium flow out of the sarcoplasm. Long periods of sustained exercise may damage the sarcoplasmic reticulum and the sarcolemma, resulting in impaired calcium regulation. It has been observed that oxygen intake remains higher for an extended period of time even after exercise has stopped. This phenomena is called the oxygen debt. Oxygen debt is the amount of oxygen needed to compensate for ATP produced without oxygen during muscle contraction. This oxygen is required to restore ATP and creatine phosphate levels, convert lactic acid to pyruvic acid, and in the liver to convert lactic acid into glucose or glycogen. Other systems used during exercise also require oxygen, and all of these combined processes result in the increased breathing rate that occurs after the exercise and known as the oxygen debt. Thanks for watching and listening. Do share this video with your friends and colleagues. If you like this video, subscribe to this channel for watching more videos.